Oh, yep, there we are. There we are. So I too have a picture of David Harvey. There's the same picture. I don't know which way round it was. You may have given this to me, or I may have given this to you, James. And of course, David Harvey was my senior colleague for many years. He was, as James has said, ahead of his time in very many different ways. It was wonderful to have someone as calm, as considerate, and as caring as one's senior colleague when you first become a young consultant. And I can truly say that David Harvey, together with Osmond Reynolds and Jonathan Shaw, are the reason why I became a neonatologist. So I'm extremely grateful to him. Now, in this lecture, I am going to, and this is not a finished product. This is a work in progress, so please bear with me. But there are three things that I want to address, or talk to you about, or discuss. The first is how we used to think about preterm birth. And the second is how we think about preterm birth now. And then I'd like you to join me in a little bit of reflection and think about how perhaps we should be thinking about preterm birth in the future. So let's go back 2,500 years or so. Hippocrates said no fetus coming into the world before the 7th month of pregnancy can be saved. That was it. It's really not something, preterm birth was not something that evolution, billions of years of evolution, had prepared for. But roll on then some 2,000 or so years later, and we have Shakespeare. Shakespeare always had something very telling to say, and he did indeed here. He represented the view of the time, and perhaps it's a view that remains with us to a little bit today. I that am rudely stamped, deformed, unfinished, sent before my time into this breathing world, scarce half made up, and that so lamely and unfashionable that dogs bark at me as I halt by them. Shakespeare's famous words from Richard III. So this, I think, has been the representation of preterm birth for many, many decades, and as I say, perhaps to some extent is with us still today. But for many, many, many years, what dominated preterm research was this image and this belief, this perception that that the uterus is the ideal intensive care unit. And I plagiarized this slide from an advert, for some of you in the audience may recognize it, from an advert for a tocolytic agent. And so this presumption, this assumption that prolonging pregnancy would improve infant outcomes has really dominated obstetric research. And over the decades, women have been subjected to bed rests, pessaries, cervical cerclage, antibiotics, tocolytics, with very little evidence of benefit. But I think also extremely important for this audience to note that long-term, meaningful infant outcomes for these interventions were really rarely evaluated. And at the bottom left there, you will see a graph taken from a paper from Jane Norman's group. This was Seminars in Perinatology in 2017. It shows the total number of research studies, those are the blue bars, the number of studies with follow-up less than 2 years, and the number of studies with follow-up more than 2 years, the green bars, for you've got on the left there progesterone, then cervical cerclage, pessaries, tocolytics, and antibiotics. And just look at the red and green bars. Tiny, tiny, infinitesimal numbers of studies that have actually asked questions about whether or not these interventions in the mother affect the baby. So when these studies were done, and there are very, very few of them, they tended to show that actually there was no impact on the baby. And in fact, some of the studies showed, I'll give you the Oracle trial, for example, showed that there was evidence of harm. So the obstetric focus moved a little bit. If there's no point keeping the baby in utero, well, what's the obvious thing to do? Let's get the baby out. So this was embraced with enthusiasm too. And, oops, sorry, what have I done? Beg your pardon. Oops. Which is the pointer? I don't want to press the wrong button again.
Underneath.
Oh, underneath it, sorry.
The bottom of the
On the back.
On the back.
Yeah.
Oh, there we go, got it. So here's a very interesting graph. This is from the Peristat group. This is 2014, and these are countries. So you've got United States, UK here, Norway, Malta, Austria, and so on. And what you've got here is the caesarean section rate by gestational age. And you will see that the lower the gestational age, the higher the caesarean section rate. If there's no point keeping the baby in utero because it's an adverse intrauterine environment, let's get the baby out. So obstetric practice embraced this new message with enthusiasm. And you will see here for Austria, for example, at the lowest gestational ages, and we're talking here at 25 weeks gestation, at the left-hand end of the x-axis, you will see here caesarean section rates of 75–80%. Let's get the baby out. Meanwhile, our very, very heroic fetal surgical colleagues have been adopting another approach, which is let's get the baby out. And then when the baby's out, let's care for the baby as though the baby remained in utero. And any of you who have heard Alan Flake talk about his baby in a bag experiments and have got to the end of the talk where the lamb emerges and tottles across the floor cannot help to be amazed and moved by how wonderful this seems. Stunning work, meticulous work, but there are 15 million preterm births each year. Are we going to put 15 million babies into bags, even if this technology takes off? This is a slide from World Health Organization in 2010, and it shows you the hotspots for preterm birth, the red areas where we have preterm birth rates more than 15%, and then the yellow areas and the green areas with the lowest rates of less than 10. But 15 million births a year, preterm births a year. And roll on to 2019, this was published in The Lancet a little while ago, and this gives you the top 10 countries for preterm births. And you'll see here that lower middle income countries dominate this list, but there is one exception, and that is the United States. So this is a problem for all of us, rich countries, poor countries alike. Of course, added to this is the fact that survival is increasing. And Mary's already alluded to this paper, but this is the graph from Shalini Santhakumaran's study of survival. You see here, you see the years here on the x-axis from 1995 to 2015. You see survival here to neonatal discharge, the denominator being admissions to neonatal units. Here we have the data from Epicure 1 and Epicure 2 studies. And then these dots are data points derived from the National Neonatal Research Database. And you'll see that for 23, 24, and 25-week gestation babies, these lower ends of the gestational age range of the babies that we care for, there has been a steady increase in survival over the years. So the numbers of preterm babies are not going down, and survival is improving. So the neonatologist's focus, always pragmatic, has been to roll up our sleeves and get on with it and look after these babies. And we've been helped when something newsworthy has come along. And for the younger members of the audience, I'd like to tell you about one such event, which was the birth of President Kennedy's son. He was born at 34 weeks gestation. He weighed more than 2 kilograms, and he died of respiratory distress syndrome. And sometime later, this report appeared in the New York Times. A cure for mysterious hyaline membrane disease, which takes the lives of up to 25,000 infants in the United States each year, was reported today at a medical meeting. A little more than a year ago, the disease was fatal to Patrick Kennedy, the infant son of President Kennedy. Doctors who disclosed the treatment declined to say, were always prudent, declined to say that they might have saved the premature Kennedy baby, But they did say that they and others who've tried the method in the last 8 months are highly enthusiastic about it. They've tested the treatment on 28 babies in 5 hospitals, and all 28 are alive today, and without the treatments, half of these babies would probably have died. What was the treatment? Does anyone know what the treatment was that they were referring to? No? No. Does anyone know? Magnesium sulfate enemas, now no longer used. And as I said, the neonatologists rolled up their sleeves and got on with it because they were being presented with preterm babies. Now, there was some shred of merit in this issue of magnesium sulfate enemas, because at that time it was felt that part of the problem of respiratory distress syndrome, so I'm told, was that these babies were waterlogged. They were fluid overloaded. And certainly I remember very clearly being told babies were water overloaded. Yes, like Neil's nodding there too. Good. And indeed, many of the babies we looked after in those pre-surfactant, pre-antenatal steroid days were indeed, well, the best word is blobby. They were waterlogged. So the reason for the magnesium sulfate enemas was that it was an attempt to draw out the fluid and make these babies less fluid overloaded and thereby improve their respiratory function. A teeny little bit of a theory behind the practice that was being evaluated, exactly in line with some of the things that Mary has been telling us earlier. Superficially plausible, but. So my own teeny role in all of this was when I was doing my MD, and this I put up to say to all of my former PhD students, all of us have suffered when we've done our higher degrees, and one of the things that I imposed on myself as self-flagellation was to meticulously measure sodium balance in babies who had respiratory distress. Now, the trainees of today will never see this. They will never see a baby who is breathing his life away, with the sternum retracting back to the back, in and out, until the baby gets more and more and more and more tired and dies. You will never see that. And that's one of the great triumphs of neonatal medicine. But this is what we were faced with in the days before antenatal steroids and before surfactant. And I had been told, as I've just said, that part of the problem was fluid overload. And at that time, there were a number of studies being done that involved the use of diuretics to try and improve respiratory distress syndrome. But it didn't make sense to me that, that we should be, well, a lot of what we did in those days didn't make sense to me. So what I set out to do in my higher degree was to meticulously measure sodium balance. Each of these periods of time is 4 hours, and I measured urine output, yes, meticulously over each 4-hour period, calculated sodium balance, and what you see here, this line, is the alveolar arterial oxygen gradient of a baby with respiratory distress syndrome. Remember, no antenatal steroids, no surfactant. And this baby is extremely unwell with a very, very high alveolar arterial oxygen gradient. And then at about, this is the 12th period, so that's 48 hours, the baby gets better. And you can see that that gradient declines. And at about the same time, these babies had a diuresis, and it's shown here by this negative sodium balance, and it's accompanied by a massive diuresis. And that was an observation that had been made clearly, but people, as I say, attempted to treat the disease by invoking a diuresis by giving diuretics, and that didn't work. That didn't have any impact at all on respiratory function. But you can see here clearly the temporal association of negative sodium balance, a diuresis, and an improvement in the baby's respiratory status. And over the next few years, that translated into a number of studies which essentially showed that, yes, loss of body water after birth is unrelated to the volume of fluid administered. In other words, we had no reason to overly restrict these babies and therefore deprive them of nutrition. And there was loss of extracellular water in both babies with respiratory distress syndrome and indeed in all babies, but the temporal relationship was changed. Also, that routine sodium supplementation, which is what we had been taught to do in those days, was inappropriate until the baby had had their diuresis, because that delayed the contraction in the extracellular compartment. Delaying sodium supplementation, therefore, a tailored approach to caring for these babies, delaying sodium supplementation until postnatal weight loss was underway, in other words, until the diuresis was underway, had a beneficial effect on oxygen requirement. And also we did some mechanistic studies showing that the postnatal diuresis was triggered by an increase in pulmonary venous return, left atrial distension, ANP release, and also we showed evidence of a postnatal maturation of an intracellular signaling mechanism leading from ANP, CGMP signaling, and sodium excretion. So that was, as I say, my small contribution way back in the days before surfactant and antenatal steroids. More recently, my research group's interest has turned to the adult phenotype of the preterm baby. This is characterized, this was our first paper on this subject in 2011, and we showed in a small number of participants that the adult phenotype was characterized by an increase in internal abdominal adiposity, even though these preterm participants were outwardly slim, very healthy, they were aged between 19 and 27, and they looked fit and well. But they had this really quite substantial increase in internal abdominal adipose tissue, almost 0.5 kilo, and they had a higher blood pressure, and this was despite the fact that, as I say, they looked outwardly healthy. And in that study, the men had a significantly different urinary metabolome. So over the subsequent years, well, we'd already shown in 2005, this was Savita Uthaya's work, showing that preterm infants at term have got an abnormal body composition characterized by increased internal abdominal adiposity. We then showed that in 2011 in young adults. We've just carried out a replication study which shows that that observation remains true. And we've now started, and some of you will have heard Emily Pryor's poster presentation, and Emily has just started a longitudinal study to ask the question, did the aberrant body composition we identified in preterm babies at term, does this persist, does this track, does this stay with them as they grow into adult life? But others have been showing similar things. So this is a systematic review and meta-analysis relating to whether or not preterm adults have higher blood pressure. And this is now a very, very robust finding indeed. The answer is yes, about a 4 millimeter increase in average in systolic blood pressure and about a 2.5 millimeter increase in diastolic blood pressure if you were born very preterm. That is a very substantial effect size. Others have shown, now you might have predicted from our phenotypic studies that preterm adults would be at greater risk of the metabolic syndrome. So this is a study from the Northern Finland birth register, and this clearly shows that that is indeed the case. So whether you look at hypertension, obesity, the full-blown metabolic syndrome, or fatty liver index, there is a dose-response relationship, and the early preterms as well as the late preterms are at greater risk. Similarly, type 2 diabetes. This is a study that shows here that the odds ratio for developing type 2 diabetes, again, you would predict this from our phenotypic studies. The odds ratio for developing type 2 diabetes is about 1.68 to 68% increase in the risk of developing type 2 diabetes if you are born before 35 weeks gestation. And then you might also predict that our preterm adults would be at greater risk of a shorter lifespan. And again, this is indeed the case. These are data from the Swedish birth registry showing that in the first 5 years, there is clearly a relationship between all-cause mortality and degree of immaturity. So you have each row here is a gestational age band. And though that goes away in childhood, and we neonatologists breathe a sigh of relief, it comes back again in the 18 to 36-year-olds. So yes, again, the phenotypic data would predict a shorter lifespan, and indeed we are seeing a shorter lifespan. And most recently, and these data are unpublished and others have also replicated this, we have shown a molecular correlate of premature aging. This is a telomere study done in collaboration with Sue Ozanne here in Cambridge. And this shows you telomere length ranges. So you've got here the shorter telomeres and here the longer telomeres. As you know, that telomeres are a marker of cellular aging, and they become shorter with age. And what we've shown is that men in general, all men, have a higher percentage of shorter telomeres. This was our young adult population, 19 to 25-year-olds. All the men had a higher percentage of shorter telomeres than the women. And they had a lower percentage of longer telomeres. But the preterm men had more short and fewer long telomeres in comparison with the term men. And in this age group, we did not see that effect in women. But of course, what we don't know is that were we to study a population of ex-preterms in their 30s or 40s, would we see this effect becoming apparent? But so we have clear molecular corroboration of the preterm phenotype. And so the emerging picture today is as follows, really. There is evidence of disruption to pretty much every organ system. And here are just some of the impacts of preterm birth that have been reported in the literature. And you can see how diverse and how extensive they are. Asthma, type 1 diabetes, type 1, this is type 1 diabetes, hypothyroidism, the vascular disorders, cerebrovascular disease, stroke, neuropsychiatric morbidity, some strange things, testicular cancer, also a lower reproductive rate, greater risk of preterm offspring, reduced lifespan, and then the cardiometabolic factors. So evidence of disruption to every organ system. And therefore, we've proposed that the preterm adult really represents a phenotype of accelerated aging. Now hold that thought, please, because I'm going to go slightly to what might seem to be a different area. Hilary Wong presented today the data from our spring study. And of course, the background to this is that 1 in 4 very preterm children meet neuropsychiatric diagnostic criteria. This is a graph taken from Hilary's paper in Pediatrics in 2014. This is an evaluation of early childhood social communication difficulties. This is the preterm population. This is a term population, and you can see the clear shift to the right in the preterm population. Now, it's not just ADHD, but also ASD, and also the other major psychoses, which are all increased in prevalence in the preterm population. And so it begs the question, is preterm birth, or its sequelae, something that directly increases the risk of neuropsychiatric disorders? Or is the increased prevalence of neuropsychiatric disorders in the preterm population an indicator of a fundamental shared underlying genetic liability that is later manifest in this way? It is clearly a key question for us. So the spring study, as Hilary's already said, will set out, because we've only completed the pilot study so far, we will set out to test whether or not very preterm neonates are enriched for rare genetic deletions and duplications, in other words, copy number variants that are already known to be implicated as neuropsychiatric genetic risks. And as you've heard today, the pilot study has now been completed. And just to summarize some of the findings from that pilot study, the frequency of de novo and rare pathogenic copy number variants appears to be higher than in a healthy comparator group. I say appears to be because I'm now entering a much more speculative part of this discussion. We've not completed the full study yet, so all I can say is appears to be. The de novo CNV identified do include those with causal effects for neuropsychiatric disorders. But it also appears that very preterm babies have increased rare pathogenic CNV compared to parents, and mothers appear to have an increased number compared to fathers. Now, the de novo mutations represent really a very extreme form of genetic variation, and they're usually deleterious, and of course there is no evolutionary selection pressure, and therefore they tend not to persist to a great extent in the population, hence they are rare. But there are data emerging now that show that the overall de novo mutation rate is increased in preterm babies. This is a study published in PLOS Genetics in 2017, and here you will see in red gestational age below 37 weeks and in blue full-term babies. And this shows the de novo mutations per genome, and again you can see a significant shift to the right. So, the overall de novo mutation rate in preterm babies is increased. Now, it's been accepted, I'm told, by my embryologist colleagues that germline de novo mutations, first of all, are predominantly of paternal origin, and secondly, that the de novo mutations found in babies are usually thought to arise through a germline mutation in one of the parents. However, other data, very recent data, have suggested that the increased de novo mutation rate in preterm babies is not only a robust finding, but also it is not explained by differences in paternal age. In other words, it may not be due to a germline mutation. And there are other data, again, this is from another discipline, another specialty, but there are other data emerging that suggest that the de novo mutations, which were previously thought to arise in either the egg or in the sperm, actually appear to have a substantial fraction that occur post-zygotically, in other words, in the embryo. Other data show that the risk of birth defects, congenital abnormalities, are increased in preterm babies. And you can see some of the numbers here. This is just one study. This is from Atlanta. This was a population study, a geographically defined study. And you can see here the relative risk for congenital abnormalities by gestational age, with quite substantial increases in the relative risk of congenital abnormalities. Hold those thoughts. I'm going to come back to them and ask you to put them together in a moment. Hold those thoughts just for a moment, but I want to give you a third consideration, and that is this, the relationship between preterm birth and socioeconomic deprivation. So there are innumerable studies around the world clearly demonstrating the association between preterm birth and socioeconomic deprivation, and we heard one this morning in relation to communication and language. This is a systematic review and meta-analysis that was published in BMJ Open, which refers to the UK. So you see here studies done in the UK, again, showing very clearly the relationship between preterm birth and socioeconomic deprivation. But we also need to recognise that there's been a widening of birth inequalities over time. And these, again, are UK data. This is from Newcastle. And you'll see here, this is year of birth. So you've got over here 1960, here 2000. And you've got the Townsend deprivation scores. So the green is the most deprived, and the black is the least deprived. And start over here, and end over here. And you will see that there has been a widening. Not only is there a clear socioeconomic gradient with a higher risk of preterm birth in the most deprived populations, but in the UK, and very likely in other parts of the world as well, but certainly in the UK, there has been a widening of this inequality gradient, with the percentage of preterm babies decreasing in the least deprived and increasing in the most deprived. And this increase in preterm births in the most deprived groups is not attributable to a differential increase in caesarean section rate, which might have been one plausible explanation. So what might link these observations? These are the observations of, let's just go through this, a bit of a recap. Prematurity seems to be a phenotype of early onset senescence, affecting every organ system. Preterm babies appear to have a higher de novo mutation rate. Preterm babies have got more congenital abnormalities. And preterm birth has got a very strong socioeconomic gradient. Let's reflect on what might link these possibly initially seemingly diverse observations. I would like to suggest to you that the fact that research to date has focused on the mother as the origin of the problem of preterm birth may be a flawed assumption. And I think that we should consider whether or not preterm birth is a fetally driven phenomenon. And perhaps even, preterm birth might be a manifestation, an intrauterine manifestation of fetal senescence signaling. A preterm baby who's aging at a rapider rate might be sending a signal to say, I am ready to be born. I did say I was entering into a very speculative part of my talk. But let's just consider what data out there might be to support this hypothesis and to suggest that this isn't as wacky as you all, I can see the looks in your face, this isn't as wacky as you think it might be. Might this be a consequence of influence impacting early and embryogenesis? Well, it is already known that parturition is signalled by fetal neuroendocrine signals and by senescent fetal tissues. Again, this is not my area of expertise, but this is out there in that literature. And there are also data from a completely different area of research, and this is the artificial reproductive therapies. That it's very hidden data, but it shows that, for example, in vitro fertilization culture medium affects gene expression, DNA methylation, birth weight for gestation age, birth weight, and, yes indeed, preterm birth. Surprising. I was very surprised when I discovered that. So are preterm birth and the preterm phenotype the consequences of implantation, of conception, implantation, and development in intrauterine milieu? The embryo culture media used in IVF affects the rate of preterm birth. But of course, there are limitations on embryo research. And much of these data many of the experiments have not been done, and much of the data are hidden. And much of the data are hidden behind a wall of commercial confidentiality. Why do I say that? Because there have been some limited trials of different culture media for IVF, randomized controlled trials too, and they very clearly show that the culture media in which IVF is undertaken does affect infant outcomes. I'd like you to read this. This was reported in Science News in 2016. Fertility experts are calling on the companies who make the solutions in which embryos are cultured during IVF to give a clear list of ingredients. This was because following publication of a trial that shows that the composition of these laboratory culture affects the outcomes of the resulting embryos and babies. Now, I wonder if you were as surprised as I was to discover that the solutions in which embryos are cultured during IVF have not been subjected to rigorous evaluation. The evaluation they have been subjected to pertain mainly to animal experimentation, and these results are not in the public domain because, as I say, of commercial confidentiality. And yet here we have an opportunity to actually understand how events early in embryogenesis may impact on lifelong health and indeed on preterm birth. This is a Brave New World scenario. So let me recap again. I think these are the questions facing us. Do preterm birth and the downstream phenotype that I've described for you share common genetic determinants? Someone today, I think it may have been you, Harvey, actually, someone today said that preterm birth is heritable and that the generally presented figures are that heritability is between 25 and 40%. But not one of these studies, to my best knowledge, has actually looked at the long-term transmission rate. These are usually only into the next generation. And you might predict a certain amount of heritability if the hypothesis that preterm birth arises as a result of aberrant embryogenesis, because, of course, a mother who is exposed to poor conditions herself is also going to be exposing her as yet unborn daughter and granddaughter to those same phenomena. I remind you too that conditions of socioeconomic deprivation also mean greater exposure in the periconceptional period to a number of factors which might adversely impact implantation and early embryogenesis. Hypoglycemia, endocrine disruptors, toxins, pollutants. These are all things that are much, much more prevalent in socioeconomically deprived populations. And therefore, my second question is, do periconceptional influences that lead to this downstream phenotype also increase the likelihood of preterm birth? Is the problem an adverse intrauterine environment? This, I think, is very plausible given the ubiquity of the systems involved. But this is a neonatal audience, predominantly, and so the next question is, does postnatal care contribute to the preterm adult phenotype? Are neonatologists making matters worse? Well, very possibly, but the truthful answer is we just don't know. Is what we do making things better? Might what we do make things better? Well, of course, very definitely. Neonatologists might very definitely be making things better. As yet, we don't know exactly what we're doing is making matters worse or making matters better. So, I suggest we need to reconceptualize preterm birth. We've learned that striving to maintain a fetus in a suboptimal environment was not very wise. All those decades of research that strive to keep a baby in utero come what may. I think that assuming that preterm birth is always maternally driven doesn't appear to be justified, given the data that are emerging from very, very diverse specialties. Focusing attention on caring for ever better ways of caring for preterm babies is clearly essential, but I don't think it's enough, because I think we should be giving some consideration as neonatologists to what are the fundamental causes of preterm birth. And so I'd like to end with a challenge to the perinatal research communities and to the Neonatal Society, and I have been a member of the Neonatal Society for many years, so I count myself amongst these who should be the recipient of this challenge. I think we should reject the premise that preterm birth is exclusively a medical issue and therefore needs an exclusively medical solution. It may well turn out to be far more an issue for society. I think we should not only be conducting research that aims to improve the care that we give babies, but we should also be conducting and promoting and advocating for research to actually understand the causal determinants of preterm birth and to understand these observations that have been made regarding the relationship between socioeconomic deprivation, suboptimal periconceptional conditions, and preterm birth. And why should we do this? Because, of course, it might be just this ultimately that leads to the prevention of preterm birth. Now, I want to end with this slide, first of all, which shows pictures of some amazing people who are all born preterm. And I want particularly to point to the fact that 3 of the greatest scientists in history were born preterm: Isaac Newton, Albert Einstein, and Charles Darwin. Food for thought. And I want to end with this slide, which is to say thank you to my wonderful research group. Not all of them, but most of them are pictured here. Many thanks indeed. Fantastic pleasure with working with all of you. And to say thank you to all of you who have listened to this lecture, particularly the 2 men in my life. Thank you very much indeed.
Neena, that was absolutely fantastic. Probably the most hypothesis-raising David Harvey lecture we've had to date.
Well, all wacky.
Does anyone have any questions, comments?
We are just in the beginning of a study of frozen egg, babies coming out of frozen eggs. I mean, the normal in vitro fertilization, I think babies are a little bit smaller than expected for gestational age. There's a prematurity issue and there's an undergrowing issue. But for the frozen eggs, there's an overgrowing issue, which is very well known for artificial in vitro fertilization in animals, so in husbandry. And obviously the question is whether this it's just the fact that they're frozen. So the culture media and all this stuff is the same. So the question obviously is this 150 grams at term age, what change will that make later on?
So I would say that I don't think the 150 grams is the issue. I think the observation is that clearly the circumstances in which an embryo is cultured does impact on what happens subsequently. And as yet, this research is in its infancy because we are confined to talking about really rather crude measures such as the 150 grams. What we really want to understand is whether or not, for example, culture conditions or the intrauterine conditions are impacting on much more fundamental processes. Hence my suggestion, given the fact that every single organ system in the preterm baby appears to be disrupted, we are clearly talking about something that is very fundamental indeed. So I offer no answers at all. I've raised only some questions, but I do think that the Neonatal Society has got a great opportunity to be inviting some embryologists, some geneticists, and some folk from the disciplines that understand this science to come together, because it's only by piecing together different bits of this jigsaw that we are likely to get a clearer vision of how we should go forward.
Yeah, this freezing issue is, I mean, the more and more eggs are frozen because there are many sort of advantages in that.
I just wanted to ask you what you think are the more important, or the drugs that we give for premature birth, you know, all the steroids that many of the women have had once or twice or more. And secondly, being born too early and not having your amniotic fluid to recycle it, and all the substance actually you don't get through your swallowing because you're born. And of course the placental hormones that you don't get either. So it's a completely different milieu.
You're right. I absolutely agree, and I think that Gordon Smith, this earlier on today, absolutely gave it to us straight about the potential impact of antenatal and postnatal steroids might have in ways that as yet we do not fully understand, but on theoretical grounds there is every reason to anticipate. So the practical correlate of that not from a basic science point of view, but from a clinical point of view, is that we really need to address the problem of long-term follow-up, not just for individual studies, but across the board. And I'm afraid that this, I think, is a real practical challenge for all of us. I think the opportunities to do this are within our grasp now. And again, Mr. President, I look at the Neonatal Society to take a leading role in actually helping resolve this problem of how we collectively solve the need for long-term follow-up, not only for the care of the babies themselves, but also for all of the trials that are being done. It is ridiculous that we should be doing this in isolation for each individual study. We should be doing this collectively. And to your second point, the final phenotype of the preterm baby, I would suggest, is clearly going to be impacted by multiple hits across the lifetime. This is the life course approach, which has implications not only for preterms, but for full-term babies as well, and the preterm baby is no exception. But my suggestion to you is that we should look for the origins of preterm birth right back at the beginnings of conception, and then, of course, there will clearly be multiple factors intervening as the baby grows older in utero and in the postnatal period and as he or she grows through life. And so there are opportunities to actually positively or negatively impact across the life course. And again, we have yet to see the proper bringing together of an emerging theory of life course science into policy and policy implementation.
Thank you for that splendid lecture, Neena, and thanks for the focus on the pre- and periconceptional period. Certainly, in animal studies, there's quite a bit that backs up exactly what you said. In the mouse, it's true that if you undertake embryo culture and then put the embryo back into the recipient female, you get many of the physiological phenotypes that you referred to: higher blood pressure, fat deposition, insulin sensitivity, and so on. It's pretty difficult to shift gestational age in the mouse, or indeed in large mammals for some reason, but certainly in the sheep, if you deprive the mother a little of food, actually gestations increase by a day or two, as if the fetus teleologically thinks, I'll stay in a little longer, perhaps to grow a little bit better, undernourish her even more, and you actually shorten gestation. So there's a lot going on there, and this is early gestation under nutrition, which fits your hypothesis very well, I think.
Thank you, Mark.
Well, if that's everything, thank you very much indeed.